Welcome, welcome everyone. I am János Juhász. This is the data visualization course. And in this block, we will talk about Gephi, a program capable of visualizing networks. This section will be a brief introduction of Gephi and then a small tutorial how to use it, about its basic functionalities. These tutorials and slides are based on the following materials, so please check, so you can check it for more details. So Gephi is a data visualization program. It's free open source and it has functionalities for network visualization and network analysis as well. It has a nice user interface and it can produce high quality images and can handle big graphs quite well. It's a popular tool for all kind of networks, so it's not biology specific. It's quite general and it has many additional plugins. Maybe if you have some specific data types or you want to perform some specific task, the basic version is not enough, but you can install new plugins with a couple of clicking. Here is the website of Gephi. You can download it from there, and if you follow the installing instructions, you can install it to your computer. Java is needed for running Gephi. You can download Java, for example, from here. If Gephi cannot find Java, you can set the path of Java manually. Here you can find a small tutorial video how to do that. And here you can find many additional tutorials of how to use Gephi in a more advanced way. Gephi is not the same as Cytoscape. Cytoscape is another program that is very popular for network analysis, and it's more biology, it's more life science specific. During the course, you will have an entire lecture of Cytoscape. Cytoscape has a broader collection of plugins related to biology, but Gephi can handle large and dynamically changing networks quite well. So Gephi is a bit more general and maybe a bit more visualization specific, while Cytoscape is much more specified for biological research. This is the user interface of Gephi. We will go through these different parts, what are they for and how to use them. The rest of this slideshow is about how to use Gephi. I won't go into details. The details will be at the tutorial part, when we actually will use Gephi. You can install plugins, like here. You go to the Tools Plugins interface, and you choose the plugins that you need from the list of the available plugin. You install them, and after that you have to restart to Gephi to start to use the functionalities from these plugins. Just the basics. When you start Gephi, you can make a new project, or you can open previous projects, and you can continue them. You can import data. Gephi can handle multiple different input data types. You can set, for example, in a text file, the delimiters, for example, comma, semicolon, tabulator, and so on. And in the importing step, you can check if Gephi understands your data right. So understands the nodes and the edges and their properties and their values right. You can check it before actually importing the data into Gephi. You can save your whole project. It is useful because in this way you can continue your work, your editing and so on later on. You can do it with the file, also from the file menu with the save as. Or you can export the constructed visualizations, again, in the file menu from the export tab. You can choose between different, you can export the graph data, or you can export figures in different pixel-based or vector-based formats. Here there are three main tabs when you use Gephi. Most of the things that we are doing in this overview tab, but in this data laboratory tab, you can study, edit, sort your data, the nodes and the edges. So you can edit the data itself at that tab. In this preview tab, you can trim the final details of the graph before exporting it as an image. For example, you can set the opacity of the edges, making the nodes more visible, or you can specify if the graph is directed, not directed. Usually directed, the edges of directed graphs are denoted with these curved lines. And the edges of non-directed graphs usually denoted by straight lines between the nodes. And if you change some settings in this menu, this is reversible and it does not affect the structure of the graph. You can set different parameters here, for example, setting this opacity, check if you want to curved or straight edges, and so on. And then if you click a refresh, this preview will be updated. And then you can export the image also from here with this button. The rest of the things are in the overview tab. At first, you can set, for example, after you loaded your graph, you can set the appearance of the nodes and the edges. You can define properties of nodes, edges like their size, shape, color, their labels, and you can customize them based on their properties. You can, for example, add the same color for every node, or you can group the nodes based on some of their properties chosen from a list of the available properties of the nodes, or you can assign some continuous value to each node and color them based on this continuous value. Here, this partitioning is making distinct categories and color them or size them based on these categories, while this ranking is assigning continuous or an ordinal value to the nodes, for example, or for the edges, and color them or size them based on this continuous scale. How it looks like, this appearance tab. You can check nodes, you want to work nodes or edges. You can send uniform color to each node. You can partition them based on here the class attribute chosen from this list. You can change with this palette menu the color schemes, the color scheme, so you can assign different colors for different groups, or you can rank them based on the degree attribute, for example, again chosen from the attribute list. And after this is the color, this is the label tab, this is the size tab, and after you finish the modifications, you click apply and the updated figure will emerge. This step may be the most important one, the layout, because it defines how the graph will look like. And if you have a huge graph, it really matters how it looks like. You have to organize the nodes in a clever way to make the structure of the graph visible. We will see at the tutorial part that the default layout makes no sense technically. You have to change it. You have many options how to organize the graph, how to organize the nodes. By default, they are organized randomly. You can rotate the positions. You can make a graph denser or looser, increase or decrease its visible size. But the more clever methods, for example, this Frunsterman-Reingold method, it handles the nodes as magnets, technically, and apply force between them, similar to magnetic force, but it will conserve the area of the graph. So in this way, if you apply that layout, the bigger nodes will repass the other smaller nodes and will require bigger space. It will make the graph a bit more untangled. While this force atlas 2 method, here you can apply different forces between the nodes, set their parameters, and it can order the nodes in a more natural, reorder the nodes in a more natural and good-looking way. Here you can select the layout, set its options, click Run. Some methods perform in one step. Some methods can run long. You can stop running if you are already satisfied with the layout, or you can wait till the application of the force result in a stabilized network. This is just some examples. So for example, and you can apply multiple layout functions after each other to get the best view. So for example, this Frunsterman-Reingold method, you can be used at first to untangle the initial random layout, for example here. And then you can use the force atlas to get the final shape. You can prevent, for example, overviews and set different parameters. And these shapes, if you look at them, these look much better than an initial totally random network. You can edit your network manually with this set of options. You can select nodes or a group of nodes. You can move them, resize them, recolor them, edit their properties. You can even add manually nodes or labels. And you can also calculate the shortest path between selected nodes. If you are satisfied with the layout, then you can fine-tune the visualization with that menu. You can set node edge labels, you can set different properties of the labels, you can set the visibility of the edge or set different background colors that can make the other colors of the nodes and the edges more visible. Or you can set a dark background color if you want to show the network on a computer screen, and a light background color if you want to show it on paper, for example. If you are satisfied with your network, you can export it, but with Gephi you can do much more. You can do different calculations and statistics on your network. These are in this statistics tab. Here we saw what this modularity and network diameter calculations are doing. Here you can select the calculation, click Run, set its possible options, then click OK, and then the result of this calculation will appear here. And then you can use the results of this calculation for further visualization tasks. For example, this modularity thing that more natural, not totally random networks, not totally random networks contain internal subdivisions called communities or modules. These are not mathematical definitions, just to get an insight what a module is in a network. It can be identified as finding these modules can be done by comparing densities of edges within the group and from the group toward the other parts of the network. So those nodes are in the same module that has a lot of edge between each other and much less edge toward the other parts of the network. So here, if you calculate this modularity, then you get a new attribute for the nodes, telling you which modularity class they belong to. So you can use this new attribute to color the network based on the module, to make the modules visible. You can do it in the appearance field. And here for partitioning, you can select the now calculated modularity class attribute. And maybe you have to change the color to make them visible, and you can apply the result of this partitioning, and the network will be colored based on the modules. Here you can see that, for example, these belongs to one module. The network diameter calculation tries to catch the most important, the most central nodes in the network. It is done by calculating betweenness centrality. Betweenness centrality is a measure that we can get by calculating all the shortest paths between each pair of nodes, and then count how many times a specific node appears in the shortest path between two others. If this betweenness centrality is high for a node, it is a clue that it's part of a lot of small shortest path between other nodes. So it's a quite central, maybe a quite important node. You can calculate it with this network diameter statistics, and this betweenness centrality attribute will be connected to the nodes. And now with the ranking, because it's a continuous value it's a value in a continuous scale assigned to each node, then you can color the nodes with this color the nodes based on this betweenness centrality by changing this attribute at the ranking panel. And here, the more dark red colors mean the colors with higher betweenness centrality value. So these are the most central nodes. So summarizing that, in the last couple of minutes, I talked about the basics of network visualization in Gephi. Please check also the connected video for more details, for a more practical tutorial. And now let's switch to that tutorial part. Okay. Now you can see Gephi. This is the general user interface of Gephi. So let's create a new project. and let's open a data file. Let's open a data file. I will use this data file. This is the same data that you use at the Cytoscape tutorial, at the Cytoscape, one of the Cytoscape tutorials. I will open that data file. It tells me that it codes an undirected graph. It has that many nodes, that many edges. It's a quite big network. And these are the values, the parameters. It hopefully looks okay. So I click OK, and this is imported to Gephi. This is the initial layout. It's totally unrecognizable, so actually it makes no sense. Here at the data laboratory, we can see what we have. For nodes, we have ID of the nodes, we have labels for them, gene ID, type, class, degree, upregulating, downregulating degree, and so on. So actually it codes, the nodes of these networks are genes or DNA segments, and the DNA and the connection between them they are upregulating or downregulating each other. Most of the DNA segments some of them are genes and some of them are microRNAs. So this dataset is about the regulatory properties of microRNAs on different genes. At the edges, for the edges, there is a source node and a target node. All the edge types is undirected. The edges also have an ID to make them identifiable. And here in this network, the weight of all edges is 1 technically. So we won't work here with the edge. We will work more with the nodes and less with the edges, because here every edge has a uniform weight 1. Okay, so this is how our data looks like. We have edges, we have nodes. The edges are undirected. The nodes code genes or microRNAs, and the connection between them is possible up or down regulation. This is the preview field. Okay, so let's go back to the graph itself. Okay, so the first clue could make the size of the nodes proportional to with their degree. Degree mean how many connections they have. We can set the minimal size and the maximal size. You can change these values. If you apply them, you still see nothing, just you see that some nodes became bigger, the nodes with more connections. Let's color the edges, for example, color the nodes based on their role. For example, they are genes or microRNAs, or we can color them with a gene ID, but there is a lot of gene IDs, so it would make no sense. The expression these are expression values. Okay, so I think color them based on their type. They are genes or microRNAs. Or we can color them by their classes. They are genes or microRNAs responsible for upregulating or downregulating. So let's try that. it shows that there's a lot of genes and some microRNAs, but still the network is way too dense. So let's go to the layout field. If you rotate your visualization, you can see it simply rotates the network. If you click the contraction, it will make the network more dense. If you click the expansion, it will be bigger, but still totally random. So let's reorder it. Here you can set different parameters, how fast the algorithm should be, and so on. You can set these parameters and you click run, and you can see an iterative process starts, and it puts these nodes at the central, nodes with a lot of high degree nodes, so nodes with a lot of edges, and nodes with smaller amount of edges tends to be at the periphery of the network. Okay, so it could run for a long. You can stop the running whenever you want. And now, if this network is a bit more untangled, we can try to apply a force field on that. For example, we can prevent overlap. It's a huge graph, so maybe some overlap is some overlap are needed, but we will see. There are gravity, scaling factors, and so on. So you can change freely these parameters, and you click run, and you see the network starts reorganize itself. If you see, it doesn't change more, so you can stop it. It's still quite unorganized, so maybe we don't need this value. No. If you don't want to prevent the overlap, then you see that the structure of the network changed quite drastically. But now it looks pretty chaotic again. You can see that the network starts to organize. Okay, so you can play with these parameters and select a network layout that is good for you. You can central the network with this tool, but it's not a good idea because you see there is some strange nodes at here. So, but most of the nodes are situated here. So if you would like to make it a bit more visible, then you can edit nodes, for example. So you can select nodes, or you can select a group of nodes. Here you see the selected nodes and their connections. You can drag a node and move it to other parts of the network manually as well. You can, for example, modify the color of a node manually. So if you're pressing the mouse button, the color can be modified. And you can make it lighter, darker, and so on. So you can size a node, So you can resize a node with this. To make it bigger and or smaller. Can color the node and its nearest neighbors. So edit its connections. You can manually add nodes or edges. You can edit node attributes, or you can calculate nearest shortest path between nodes. In this network. Then here in this tab, you can adjust the visibility of the edges, or you can make the edges disappear totally or appearing. You can show or hide labels. Here it's clearly overcrowded with labels. You can set the background to darker or to lighter. It depends on where you want to show this visualization. Or here in this more detailed section, you can change the background color. Of the whole image. Or you can define the color of edges. For example, with that you can make the edge with the same color as its nodes. You can make them less or more visible. With the labels, you can set labels for the nodes. You can define their color, define the size of the labels. Same you can do with edges. And for example, the node labels can have the same size, proportional size to the node itself. Proportional size of the node itself. With these configurate options, you can choose what you want to show as a label. So for example, you can choose the class and clicking OK. We will see the same labels as that was the basis of the coloring here. Technically, I'm still not satisfied with the layout, I must admit. So let's increase the scaling factor as well. Maybe it looks better. Let's let overlap. Yes, now it looks better. If I set the scaling higher and I let overlap because I have a lot of nodes. Okay, now it looks better. That at first I increased the scaling parameter, and then when the nodes were more or less organized, I prevented the overlap and it separated these nodes. Actually, because of the nature of the data, this is gene regulation data, and microRNAs regulate a lot of genes. So here, these central parts are the microRNAs, and the small purple-like nodes are the genes themselves. And you can see that one microRNA regulates a lot of genes. So we can see it in this network. So here, if I select one node, I can highlight it and its connections. Because if you see that, set the edge visibility higher, so you can see that one microRNA regulates a lot of genes. And these genes regulated by microRNA are usually organized in the same group. So here in this network you see less gene regulation and more microRNA gene regulation. Okay, so let's do some calculation with this network. So let's calculate this modularity. Click OK. It starts calculating. It shows some modularity classes. This is a general report after the calculation. You can also export these images. You can save it if these plots, if it's these your interest. But what we try to do here to color the nodes based on their modularity class. It sees we have 1, 2, 3, 11 modularity class, but this palette use only 7 colors. So let's change it with this palette option. Here there are predefined color schemes. They could all gray or white, but you can also change this palette, increasing, for example, the name number of the categories. We can increase it to 11, for example, and you can choose from different color schemes, and clicking to generate, it generates more and different colors. And if you click again, you get a new color scheme. Let's say we like this color scheme, or you can choose another. It shows pastel colors. You can choose bluish colors. It's again important. It is again, you have to consider where you want to show the figure on screen or at paper. Reddish colors. So you have a lot. These fluorescent colors. Okay, so let's go back to some. It's ugly. Some more conservative colors. Let's say these, for example. Then you click OK, and you can apply these new colors to the nodes. Sorry, it doesn't look good. So let's go back to the default ones. Okay. Okay, it's better. Okay, so you see that these are the nodes belonging to one modularity class. And if you see, they are more or less they are more or less the nodes regulating by the same microRNA. Maybe I change the edge color to be a bit more darker. Okay, yeah. So here, for example, these belong to one module, these nodes. We can try to identify the more central nodes, calculating this network diameter. We have undirected graph. It's okay. It works, it calculates. If you change the parameters of the modularity calculation or this network diameter calculation, definitely you will get different results. You can play with these parameters. Feel free to play with these parameters. Here you have the general statistics of the calculation. And now with on the attribute list, this centrality measure appears. And you can color the nodes based on their centrality. Here you can, for example, invert the colors, having the higher values lighter, the lower values darker. Maybe setting the smaller nodes darker, it's a good idea because they will be more visible in this way, and the big nodes are already visible with a light color. You can choose different schemes, apply them, and so on. You can choose the range of the colors, for example, to this. So, for example, here we will have more darker colors because we increased the amount of dark colors in our visualization. Here we have more light colors. So you can fine-tune the color scale as well. You can fine-tune the color scale as well. Okay, the previous one was definitely better. So you can modify your color schemes and so on. And if you are satisfied, you can export your graphs in some format. So for example, you can choose in PDF, SVG a vector-based format, PNG a pixel-based format. For PDF, it still have high resolution, but can be open also with a PDF reader. You can name it one, and you can save it wherever you would like to save. Okay, so that was a basic tutorial of how to use Gephi. There are much more tricks, much more parameters that you can try. So please feel free to experiment with the calculations, with the algorithm layout parameters, and with the visualization tricks. Thank you for your attention.